Welcome, everybody. My name is Jennifer Hall, and I serve as the Chief of Data Science for the American Heart Association. And I'm here with a number of my colleagues to tell you a little bit more about some exciting research, data, and cloud-based platforms and workspaces that will help you drive your research, apply for grants, and provide some innovation. Chris, can you take me to the next slide, please? Sorry. Thank you. So today we're really focused on this amazing research data that you have access to that we're going to tell you a little bit more about, and workspaces and statistical analysis tools, as well as others that are available today. Here's the team. Again, I'm Jennifer Hall. I have Pratik Malia with the American Heart Association with us, as well as Brianna Woods. And then the team from NHLBI or Valsera, which is Jack, DiGiovanna, Yasemin, who's going to provide a use case for us today, as well as Ambrish, who's going to also provide a use case and give you an understanding from a researcher's point of view or a clinician's point of view, how to really utilize these tools to your advantage. And I also want to mention Natalie as well with Biodata Catalyst. So a little bit of a preview in terms of the types of data that you can access. So as you know, cardiovascular data comes in many forms and many facets. And you can see here on this slide that there's a lot of genetic and genomic type of data. There's single cell data, mass spec, imaging, demographics, clinical real world data, and environmental data. And we'll give you a pretty good sense of that today in terms of, depending on the type of data you want, you may want to access either the American Heart Association and or NHLBI's Biodata Catalyst. So here's just a preview of the agenda. We're going to pass it over to Jack, and he's going to provide an overview for all of you on Biodata Catalyst. We're going to go then to Dr. Sufi, who's going to do a research spotlight on Biodata Catalyst. Then Pratik will give a precision medicine platform overview, again, the AHA tool. Then we'll talk a little bit about open access and bringing your own data to these workspaces, which is another option, many options. How do you get started? We'll then turn to our second research spotlight if we have time, and then take any questions. So with that, I am thrilled to turn it over to Jack, who's the Chief Science Officer of Valsera, and he will provide an overview of NHLBI's Biodata Catalyst.
Thanks so much, Jen. Really excited to be here. I want to also credit Zelia from our team, who was really key to getting these slides together and showing the overview. I wanted to zoom out a little bit to Biodata Catalyst overall. It is a vibrant data science ecosystem that's actually built by a number of different leading partners in the space. From my perspective, highlighted here in red, is the part of analyzing data. So I'm the co-PI that's representing Biodata Catalyst powered by 7Bridges, the analytics platform. So we'll dive in there, but I really encourage you to check out BDC overall at the website that's linked here on the slides. So you may be asking, well, why an ecosystem? Why are there so many different components? Well, research is complicated, as you all know in the audience here. And we wanted to give this one example of Dr. Phelps, and he's interested in specifically something around lipids. He has a hypothesis that he wants to test, but he doesn't want to go across, all 85 plus individual top med studies. He wants to use something like a portal to be able to go through and explore the variables that are matched to his hypothesis, figure out what cohorts are available, and then bring that data over to an analytics workspace. That workspace would be compliant with the NIH GDS policy, requiring specific NIST certifications for the data. Also, his collaborators could be there, and his own data could be there. And then from that workspace, to be able to analyze that data, run secondary, maybe analysis, differential expression, different things, and then do further analysis and tertiary analysis after that, and then eventually get to the results that lead to a manuscript. So I want to dive in the next slides of a couple examples of what that exploration might look like. Here is one that's developed by the Gen 3 team at the University of Chicago. It's nice here. You can see the faceted search on the left, that it's aware of the access that the user has. You can see the different lipid variables that are being considered. 7 projects, about 5,000 subjects, and there's some demographics of the folks of what is that 5,000 subjects looks like. And it's button push. You can then push this into the workspace to be able to further analyze the BAM files, for example. Similarly, the picture system, you can go in and really search the different phenotypes. You would again be looking for lipids here. You're looking for, they have diabetes in this case. They're able to do both faceted and text-based search. And in this case, what's nice is you can send over again those raw files that are associated with this data, or you can package up this data and put it right into a data frame for further analysis in something like RStudio or Jupyter. And within that workspace, which also really exciting, is the collaboration that it enables. So we didn't want to design Biodata Catalyst or Biodata Catalyst 7 Bridges in particular just for the developers who are comfortable working in a terminal, but also to be able to work across their team so she can collaborate with her colleagues that are more no-code or low-code users, so that administrators can go on and understand the costs and billing and monitor and a variety of different things. So you're able to bring this whole team together to be able to drive things forward. And then once you have, oh, sorry, I used the down arrow instead of the click. Once you have that all together, you have your collaborators, you have the data that you're interested in. A really powerful feature of the platform is multi-cloud, so you can analyze data where it lives. While a lot of the top med data is available on AWS, there are other important research data sets that are only available on Google. There are some that are only available on Azure. So it's helpful to be able to move the analytics to where the data is and not have to deal with egress. More importantly, as data access committees are evolving and policy is evolving, there is increasingly a stance of enclave being required, where the data can't move. Even if you were technically able to do it, you need to be able to ensure that the data stays where it lives. So that's really critical to have that capability. On top of that, on the sort of secondary analysis size for tools, workflows, pipelines, there's over 900 of them. This grows all the time. I believe that screenshot was from August. But you have all these tools that have been developed by users over the last decade that have been optimized, that are available for public use. Further, you have interactive analysis with SAS, Jupyter and RStudio, so that you can complete the cycle there. And this is really interoperable by design, leveraging APIs, GA4GH, and other international standards. There's a link in the slides so that you could actually do that import from picture, which I showed 2 slides ago, if you wanted to take those variables and put that into an interactive analysis environment. Now, with great power and capabilities also comes great concern about cloud cost. This is something that we saw when these platforms were first coming online in 2015. We still see it today, but we've learned over time by really working with the community of what can we do to help people to better monitor and control those costs. The first thing, which is shown in the middle here in the quick pop out, is having so many people who have run these analysis over time with different size data sets. We were able to make machine learning based estimators to predict your cloud costs, so that you can take a look before you do a run and say, oh, this should cost about $3. It should take about 2 hours, whatever that may be. Further, we're able to control costs with billing groups, so that you could say you could spend up to $100. If you spent more than that, new tasks won't start, and you'll get notifications that, hey, you're about at your limit. On the bottom right here, we're showing the tertiary analysis. In this case, they're running 3D Slicer, and they have selected GPUs and the instance type. Directly underneath that, you can see that it's going to cost $3.06 an hour. And by default, we have a suspend time on here of 22 minutes, so that if I go to get a cup of coffee and get distracted by something shiny, this won't just keep running and accruing costs, but rather it will automatically save the state and turn it off, preserving that environment for when I'm back and ready to work with the system. We've also partnered with the NIH Strides initiative, such that we can offer the same discounts that are offered for researchers there by the different CSPs. And we have a way for academic users can get started with $500 of credits to try the system and move their research forward. With that quick overview, I'm delighted to hand it over to Dr. Sufi to be able to talk through one use case of what was done on the system.
Hello, my name is Yasaman Sufi, and I worked on developing WGS to IBI, which is a pipeline for genomic data analysis. I built this on Biodata Catalyst on 7 bridges, and the reason I did that was that it provides all-in-one access to from data and authorization and data like TopMet cohorts from Gentry or phenotype and other covariates from Picture. It also has public tools like Playing, Genesis, and many other public tools available. And it also provides resources to build your own and customized apps and workflows, and also provides a scalable and computational resources and storage. This is the data browser section of the 7bridges platform, and here I have searched JHS, which was a data that I wanted to explore in my analysis. And I needed the whole genome sequencing data, and the challenges for WGS data is that they are large and they are usually controlled access and they need authorization. And BDC made it very easy for me to search and access the data and copy it to my own project. Here I searched JHS cohort, and I have filtered JHS to the latest version, Freeze 9 VCF formats that I needed. And these files are very large, so it is not practical to store and manage these files locally. And on BDC, these files are, as Jack explained before me, they are on the cloud available, and they can be copied to your project for analysis, and you don't pay. That is my understanding, that for these files that are originally control accessed and already on DRS system, you don't pay storage cost as well, which is a bonus. The platform also includes many public apps like Playing, Genesis, other pipelines for quality control. And these apps could be copied into your project for your own analysis. And it also provides the option to create your own app. It can be either a single command line tool or a multi-step workflow. And it is compatible with CWL language, which makes it easy to bring in tools you've already developed elsewhere, or migrate the tool that you develop on BDC to other places. And it provides a visual editor and a code editor, so it is flexible for both beginners and advanced users. This is the public resources section of 7bridges that includes several public projects. Public projects are basically case studies or demos of tools and workflows, and it's a way to get familiar with how to combine different apps and workflows in real studies. And here is highlighted my WGS to IBI project that is published here. So this is my pipeline, WGS to IBI, and the purpose of this pipeline is to analyze genomic data and apply individualized Bayesian inference for genome-wide association studies. It includes pre-completed tasks to explorers and ready to reuse. This is the file section of the project. I copied the VCF files, VCF genomic files that were needed for this analysis into this space. These are the actual data inputs that are fed into the pipeline. This is the app section of the project. Here you can see the list of custom apps that I built for my analysis. These apps are streamlined and designed for WGS to IBI pipeline. And in the next slides, I'm going to highlight two of these tools, GD search and VCF to SNPs. The GD search is like a single line command tool, and VCF to SNP is a modular workflow. This is the user interface for GD search and an example of how apps look to users. Tool clearly ask for the task inputs, which are the input files and parameters and anything that the user should provide prior to using this app. And this is the execution settings. And user can easily select compute resources from AWS instances based on their own CPU, memory, or GPU needs. And so the user can browse from the available instances, and based on their needs and the cost of the instance, they can select the computational instances. And it also makes the scaling the analysis pretty straightforward. This is the app developer side of things. When you want to develop a custom app, this is the visual editor, which is perfect for beginners. Here you can define inputs, parameters, outputs, and under the hood, it's just running a Python script that you can see here. So Docker files are also used to ensure that all packages and dependencies are included. So to put it simple, you have an algorithm, you develop a Python code or any other code for your algorithm. And here is where your algorithm and your code is turned into an app, which can be more easily reusable and scalable. This is the YAML code for your workflow. And this is like the workflow definition. And this makes the app portable. So you can develop the app here and transfer it outside BDC too. And it ensures transparency and reproducibility. This is the other tool, VCF to SNP. This is a workflow app, which is consists of it's like chaining tools. It is a streamlined, and the data has a flow from the VCF input to the pre-processed genomic matrix, and multiple apps are used in it. This is the task section of the public project, and in this section you can see the pre-completed tasks, and it also includes run cost and time, which helps for estimating the cost and time that is associated with your own analysis. And I think in general, public projects are like this are good for demonstrating the application of your algorithm. And it's a good way to share the performance of your results. And it is also a good way to promote your algorithm with a tool that research community can use more easily. This is the data studio of the project, and this is a good place for interactive analysis and analyzing the stats and visualization. Here I have Jupyter notebooks for post process analysis, and so this was an overview of my experience with BDC platform, and I think WGS to IPI, my pipeline, makes WGS analysis reproducible and scalable, and BDC provides all-in-one access to data, tools, workflows, and computational resources and everything. Also, I want to highlight that their support is excellent, and they helped me from the very beginning. I wasn't very familiar with developing workflows, and they guided me in transferring everything, my code, tool, the data that I needed, and everything from different platforms to here on BDC 7 Bridges. That concludes my part of the presentation. Now I'll hand it to Parthik Malia, who will give an overview of the precision medicine platform. Thank you.
Thank you, Dr. Sophie. Hey everyone, my name is Pratik Malya. I'm part of the data science team at the American Heart Association. I'm going to give a brief overview of the precision medicine platform, the data sets available, and how it might help facilitate your research. Simply put, the PMP or Precision Medicine Platform is a cloud computing platform and a data marketplace that features AHA datasets. It allows teams to easily collaborate, securely access and store data, and utilize computational tools for analysis and research in virtual workspaces. We have investigators, researchers, and grantees using the PMP for analyzing AHA data with hundreds of papers published on Get to the Guidelines registry datasets and over 500 publications from our AHA grantees. We've had increasing participation from researchers doing self-analysis on the PMP as well. We have also hosted several data challenges and data boot camps utilizing the PMP. Our Get the Guidelines registry data is comprised of over 14 million patient records across nearly 3,000 US hospitals, with 55% of US hospitals participating in one or more Get the Guidelines modules, providing access for over 80% of the population. Like I mentioned before, we have had over 800 publications stemming from this data set. The PMP itself serves as a data marketplace for all HA datasets. You can find and request access to these datasets in a secure workspace. Please visit pmp.heart.org/request for a comprehensive list of all the datasets available, or follow the link in the chat. The PMP plays a crucial role in furthering AHA research goals by improving scalability, discovery, and impact, while also providing collaborative infrastructure and access to high-quality data and real-world evidence generation. We've highlighted some of those areas here on the slide. A brief overview of some of the platform's features. Please use the link in the chat again for more information. The PMP portal allows users to submit dataset requests, access documentation and support, create and share workspaces with their team, and adjust workspace configurations. While the cloud computing workspace allows users to access datasets and tutorials, write code, install analytical tools. The platform supports the usage of JupyterLab, RStudio, and SAS Studio, and users can seamlessly switch between multiple applications as needed. Each workspace also comes pre-installed with a set of commonly used packages for data analysis and modeling. Users also have the capability of installing other packages or libraries if needed. We have several opportunities for people getting interested in involved with research. We host data challenges, we host data boot camps and research awards on the PMP. The data boot camps are training opportunities for PMP research hosted by our AHA data science team. We have the next one coming up in November for scientific sessions. Please find more details on our webpage or from the link in the chat. We also have grant opportunities listed on the page as well. You can view these on the PMP webpage by either navigating to the research tab or by following the link in the chat. I hope this was an enlightening overview of the PMP as a whole. I'll now turn it back over to Jack to discuss the open access features on both platforms. Thank you.
Thanks so much, Pratik. And this actually comes to some of the questions that we had in the chat that we were trying to answer. Really appreciate the questions that you all have, whether it's in the chat or the Q&A functionality. So how do you get access to these systems? And something that came up already was, well, what about these data access requests, whether that's for Get with the Guidelines data or TopMed data or whatever other data set that you have? Both of the platforms, we have links here in the docs that help you submit your data access request, format and submit it, connected to the appropriate parties to evaluate that. I want to be crystal clear. We are the platforms that support this. We do not do the data access on our side. There's no fast track or anything, right? It's all governed through dbGaP. So that's the way to get research access to the data. But we try to help with as many resources as possible for people to understand how to do that and how to move that forward. And one thing that people commonly say, and even though in my experience, dbGaP has been getting much faster over the past year or two, well, hey, this takes too much time. It's challenging. What can I do in the meantime? Well, in the meantime, you're able to already get started in a couple different ways. So there are 2 synthetic data sets in Get With The Guidelines, one for heart failure and one for stroke. There's the 1,000 Genomes Project. You can link to any open access data on the entire sequencing read archive. You can import from generalist repositories. These are all open access data sets. So you don't need approval for them. You can go in and use them directly. And then also on both systems with the PMP in red and the Biodata Catalyst in blue here, there's a GUI-based upload for files that you have locally. You can do multiple files. We can bring in files through the data repository service, manifest. We also have this concept of volumes, which would allow you to link to your files on your S3 bucket or wherever they live, so you can analyze them on the platform without actually moving them. So this is actually a lot of the way that people use the system today, is to bring your own data flow, and both platforms support that. And there's also open access data that you can get started with. And we're really mentioning a lot of times that getting started is easy. One of the ways that both of our platforms is measured is how many users we have and how active those users are. So that's the call to action is, hey, why don't you check out an account today? You can get it done before we finish Q&A, I bet. It's quite straightforward to do. Again, linking here to the different docs that help you get through this, how to get a PMP workspace or how to get an account on Biodata Catalyst powered by 7 Bridges. You could get this done very rapidly. There's extensive docs throughout, maybe not just creating an account, but everything else. So starting from the left here, there's the PMP user guide, which has a link going counterclockwise. You have the overall documentation for the entire Biodata Catalyst ecosystem, including all of the components. And then there's a more focused documentation specifically to Biodata Catalyst powered by 7 Bridges on the top right. And you may be saying, well, I don't want to read all those docs. That's too much. What if I just need help? What if I need to reach out? Well, we've got you covered there too, with extensive support teams in both organizations. On the left here, you can see the PMP UI so that you can tell them who you are and what question you have and send that over. On the right, we have similarly a context-aware support that's coming from this little question mark on the bottom right of any screen. And it tries to guess what you're doing. This is coming from a page that had a lot of projects. So it's in the top, you can see the links for, how do I find files? How do I create a project or view a project? But also on the bottom, you have a blank space here where you can look in and say, well, I thought I knew this platform all too well, but when I log in today, everything had changed. Send that over. That's going to go directly to the support. They'll know who you are. They'll know where you were on the platform and what you were doing, and then try to do that to help you find the best path forward. I think, yes, Dr. Sufi showed an exceptional example of getting something into a public project. This is really important, not just so that you have the tools and have the docs, but you have this sort of really nicely formatted warm start that has the data that you need, has the tools, has those tools configured, tasks that ran that are successful. And you can do self-guided onboarding in that way. We talked about all the workflows that were available a few earlier in the section I did before. Importantly, these are all modifiable too. It's not that you have lockdown workflows that you can't do anything with, but in this case, you're showing a graphical editor where they're adding FastQC analysis. You could also bring in tools from great repos like DocStore. You could bring in from whatever Git repository that you have, being able to bring the tools in that you need, whether that's in secondary or tertiary analysis, and create your tools as well. There's also office hours. I'm going to show them for both teams here. And at the end, we're going to present a joint office hour that we're going to do after this webinar until the next webinar. But the PMP team does this Tuesdays and Fridays, gets a lot of people in, can sometimes break into the weeds for different questions. You can come by, no pressure. Similarly, for the Biodata Catalyst 7 Bridges team, we do these on Tuesdays and Thursdays. It's nice to see that you're able to work with the community engagement team. That's fantastic and can really help researchers. What I like even more personally is seeing sometimes when you have 2 different researchers in there and they start helping each other. Oh, did you consider this tool? Did you consider that? So lots of links and things to go through here and leverage. And with that, I want to turn it over to Dr. Pandey for his use case with the AHA Precision Medicine platform.
Hi, everyone, and thanks so much, Jack, for your presentation. And so today I'll talk about my experience with working with the AHA Precision Medicine platform with an example of a project that we did focusing on developing a novel machine learning-based risk score that incorporated social determinants of health to predict outcomes in patients hospitalized with heart failure. So this idea was focused on addressing the knowledge gap that existed at the time with our approach to risk prediction, where the Get to the Guidelines had a heart failure risk score that was using logistical regression model for predicting risk of mortality, and it incorporated race as a covariate and did not account for social determinants of health. And this was problematic because it was not completely capturing a lot of the drivers of higher risk of adverse outcomes in socially disadvantaged race groups. And our hypothesis was that improved and more equitable risk prediction can be achieved when risk is assessed using ML models that are agnostic to race or that are race specific and account for social determinants of health. So to do this project, we linked the Get to the Guidelines heart failure data set with social determinants of health data using the residence zip code data that was available in the Get to the Guidelines heart failure registry, and linking it with some of the publicly available data sets on social determinants of health at a zip code level. So the analytical workflow for this project was as follows, and this was all done using the AHA PMP platform. So the Get to the Guidelines data was stored in a SAS database format, and we had around 1.8 million patient records available in the Get to the Guidelines platform. We obtained the zip code level social determinants of health data from the American Community Survey and US Census Bureau resources. We imported and linked the data using the zip code, and the analysis was done using R in the precision medicine platform. And obviously, all this was supported using the AWS cloud network, and the specifications of the network, the cloud space that we used is shown here. So this is just an example of how we linked the data, and we imported the data from the Get to the Guidelines SAS database, and all of this work was done leveraging the available workplace in the precision medicine platform. And it is really nice because you can use R Studio if you're an analytical person, or a SaaS platform if you prefer using SaaS as your main analytical software. And that's just highlighting where you access the different platforms. So they're all available through the Jupyter Hub, which is the workspace that we used. And the Get to the Guideline files are available that we can pull up, and then basically we link it, and then we can import the data in our workspace, which was using the RStudio platform. And then we can write the code, and we were able to generate the outcomes of interest, and the outputs are shown, and it's all very intuitive in the workspace when we are coding the analysis. And then you are able to really create nice graphs, and these are the figures from our paper that was published, highlighting the data outputs that came directly from the PMP platform workspace. And we were able to do this study, which was leveraging a lot of the existing data and Get to the Guidelines, but also bring data from outside to link it, and then perform a lot of the high-end machine learning analysis. And this was a collaborative effort, and AHA and Jennifer Hall were really instrumental in providing us the resource support through the AHA PMP platform to get this analysis completed, and we ended up with a very high impact publication in the JAMA network, JAMA Cardiology Journal. So we really liked several aspects about the PMP workflow. The PMP uses AWS as a backend server, which is really powerful for processing. The technical support we received from the AHA staff was really excellent and prompt, and it was really intuitive to export analysis files, and really easy to export figures that we generated. I think at the time we had some, I guess, overall things to look forward to, like posting codes on GitHub, which was not available at the time. Obviously, we had all the Get to the Guidelines data available, but more it was not available at the time because of restrictions in how Medicare data can be used. And I think when I was working on it with my analyst, we had to work on it, I think, sequentially, because real-time shared analytical space was not available at the time for that. And some of this may have been addressed as the PMP space continues to evolve and become more, I think, tailored for collaborative research analysis. So I think overall, I would say this was a really great experience, and we were able to get really complicated and analysis accomplished in this PMP space for our project. And now I'll hand it back for any Q&A, and happy to answer any questions.
Great. Thank you, Ambrish. Great overview by everybody. And now we're just happy to answer some questions. So if you want to put them in the chat, I know there's some already there that we'll start with. And or you can put them in the Q&A at the bottom of your Zoom link there. Any or both are available, but let's just start with some of the questions. So Lorenzo, you've asked a few questions around, is there a cost for the PMP and others? And so I can take that one there. For academic researchers, the American Heart Association provides research credits to cover the majority of the cost for a limited period of time. So that's we're happy to take questions. If you want to just send an email to me or Pratik, I'll put our emails in the chat and we can address any questions that you may have that are a little more lengthy on cost. Lorenzo asked, out of curiosity, why don't you have MLR3 for R? Any takers on that? Can I ask, are you specifically referring to the PMP or the biodata catalyst, or both? If you can put that in the chat, that would be great. There's another question. Go ahead, Jack.
Sorry, just to clarify, Lorenzo, is that the package? I just had to do some quick Googling.
Yeah, let's have him put a little more information if he can. That would be fantastic.
Okay.
And yes, go ahead, Jack.
I see it's a machine learning package. Thank you very much for the feedback. I think both of our systems look at feedback like that and consider it for our backlog and roadmap. If there's enough interest across researchers, that could be something we do in the future. But if it's not there today, yeah, we just haven't heard it enough as a request, at least on the BDC side.
And on the PMP side, I can answer as well. So the package list that I shared is like the set of default packages that comes pre-installed on every workspace. But this does not mean that you are limited to those set of packages on the PMP. You have the capability to install any additional packages as you see fit. So if need be, you can do that. Yeah.
And I'll just add that we were able to do so and bring like our packages for lifetime risk assessments and all, which was not part of the initial R available, but we were able to add on plugins for other packages to run analysis and to create graphs, particularly from like lifetime risk estimation and all, which is more complicated.
Okay, thank you. Wonderful. So I'm going to start at the bottom now and work my way back up. So we have a question: are there ways to trim down the variables you need and can export into CSV like Dr. Pandey did, or do we send the entire data set? Pratik, do you want to take that, or Amber?
I can take that, Jen. So it is dependent on the sort of project and the proposal. In a lot of cases, we do give you access to the entire data set as a direct connection with our data lake. So what that means is you can interface with the tutorial notebooks that are available, and you have access to the data documentation as well from the portal page. So if you were to look at like a specific subset of variables, you can use the API to filter down the specific variables you need. Or if you would rather like to have the entire data set as a whole, import it into a data frame and then begin your analysis, you can do that as well.
Wonderful. Thank you. A couple of questions regarding biodata catalysts. So maybe this is for you, Jack. There are many NIH-funded trials like TopCat, AllHat, MESA. Are the majority of them available here? Also, do we have to request approval separately on MESA?
Yeah, so yes, there are many different trials that are available on Biodata Catalyst. Zelia can probably speak to that better. I also had in the chat that there is also through interoperability, which we didn't really show in this webinar. You can also, for authorized researchers, if you had access to, say, GText, you could bring that over. If you had access to Target or Kids First or whatever other data set was appropriate for your research question, you could bring that over to Biodata Catalyst as well.
And I'll just add that if you're using data through Biodata Catalyst or BioLink, you don't necessarily through the NHLBI's data repository. The data base may differ a little bit if you go through MESA and get access to those data directly versus what you have available on BioLink and Biodata Catalyst. But the purpose of Biodata Catalyst and BioLink is to help researchers gain access to these cohort data independent of the coordinating center.
Thank you. So another question, can one extract data? And so it'd be helpful here, Lorenzo, if you can talk to us about what platform you're talking about. Can one extract data from the files directly and just write the results locally? Pratik, do you want to take that?
Yes. So on the PMP side, we do not allow extraction of data outside of the platform unless it is data that you yourself uploaded into the platform, as in which is either like a data set that belongs to you or a data set that is publicly available. In which case, our support team can work with you to get those data sets out of the platform. Typically, we expect all analyses on AHA data to be done in platform only, and we do allow you to like export the results of your analysis out. And by results, I mean any sort of like report that you may have written, any sort of visualization tables that you may have generated as part of your analysis that you need for development of your publication, that we do allow export of.
Wonderful, thank you. And the reasoning for that is that we want data to be accessible, but also private and secure. So that registry data is American Heart Association data, and there's certain governance rules around it and sharing it. And so that is why we allow you to access it within the platform, but unable to remove it from the platform. But all the results that you generate can be removed. So thank you for that, Pratik. Let's talk a little bit, everybody, I'm going to just go around the horn here about, there are a lot of people asking if they can use PMP and Biodata Catalyst together. And this is where there's some challenges really within the current system. And that is because for AHA data, as we just mentioned, you cannot take it out of the platform. If you bring your own data to the PMP, yes, you can take that out. But it would be difficult to get a TopCat data set, let's say from Biodata Catalyst. But let's turn it to Jack and see his thoughts on how people might combine data.
Yeah, it's a very exciting topic, and I think fundamentally these decisions of whether or not you can take data out or not, or download data, this is coming from above, from the NIH genomic data sharing policy, and also from the individual data access committees for each of them. So for example, as Jen and Pratik said, if you brought your own data, that's perfectly fine to download. If it's open access data, that's a different thing. It's kind of hard to paint with just a broad brush of things. But in general, it's a similar approach that you should not be downloading data from the system, largely for the requirements that operates within a NIST 800-171 environment, and that the data could also be sensitive. If it's downloaded, if it's on the system, every time you access it, we're going to check and say, oh, Jen still has access to this data set. Cool, let her run this analysis. once you've downloaded it, there's no more control or auditing of the data or understanding what's going on. And there's just it creates a large risk for the data owners. And so that's why these controls are in place. In terms of combining, though, there are what's called ISAs, interconnect system agreements, where you could say there's two different platforms. We do not yet have one of these between PMP and BDC, but just there are other platforms that we do, where you could say it's okay to bring data between these two systems because I trust both of these. These two systems are both trusted, and it's considered as one environment. That's a long process, but it is something that we have with the Broad's Terra platform, for example, with University of Chicago's Gen 3 platform,
Yeah.
Kids First portal, right? So there's a lot of reasons why you could have to do this. It's just it's not something you really do on a dime, like, hey, we're just going to spin up an ISA this afternoon. It really, we'd have to say there's a compelling research case. We would love to find researchers who want to leverage both, get with the guidelines in Framingham's, for example, but then there would be some work between our two organizations and between the relevant DACs to make sure that that's okay.
Great answer. Thanks. Anybody else want to address that question or experience? Okay. Let's go. Dr. Sufi, I'm going to ask you a question in the chat. You were talking about the VCD files being large and in the cloud, and this participant is wondering whether they can use an input the files on the cloud directly, and of course, write all results and log in my local storage.
Okay, I think they're talking about the VCF files that I showed.
Yes.
So the VCF files that I showed are from TopMed, and as Jack just explained, this specific data that I showed has controlled access and needs authorization. So technically, the user cannot download the data and run it locally. And I understand that that is the question here. So if the data is not controlled access and it is your own data or public data, it could be downloaded and you can run it locally and store it locally. But BDC, from my experience, is providing a platform that you can easily access the controlled like TopNet cohorts that are stored on the cloud, bring it easily to the platform to do the analysis, and then you can save your analysis results and publish them if you want.
Zelia,
I hope I've addressed.
that very good. Thank you.
Yeah, I wanted to add to Dr. Sufi's answer. Actually, one of the reasons that we highlight, and Dr. Sufi and Dr. Giovanna highlighted all the apps and the workflows, and our studio is particularly because of this, right? Because the data cannot leave the system, that's why we provide a way for you to bring your scripts, your tools, and use the tools that are already available that can really get the research done, right? So if you're looking to do something, instead of bringing the data out of the system, try to bring the script into the system or the tool into the system and work with us to make that a sort of an easy way. I think that's one of the reasons Dr. Sufi was talking about. She wasn't as familiar with the cloud or how to run a workflow in the cloud. That's where we can really help cross those two dots. And it's the same thing for the PMP.
Thank you. That's an excellent answer. And again, it's what Jack said at the beginning. We're interested in bringing researchers to the data versus taking the data and disseminating it to all researchers. So that it's kind of a shift in the way we're thinking about this, but it does give you the computational resources to be able to analyze it in one single location.